Bismillahirrahmanirrahim. This lesson consists of unit number 17, inheritance, and subunit inheritance as well. Because this topic, inheritance, unit number 17, consists of four subunits, subtopics. First one is variation, then the DNA, and then inheritance, and then selection. So this topic is quite lengthy, and you are supposed to bear me up for a longer period of time. So first subtopic in this lesson is to describe inheritance as transmission of genetic information from generation to generation. That how the transmission of genetic information can be transmitted, can be transferred from one generation to other, and even generations to generation. So the inheritance is a process by which genetic information is passed from parents to offspring, yes, offsprings. So this information is carried in the form of genes, which are section of DNA, means segments of DNA found on chromosomes, because chromosomes are the physical structures having DNA and proteins and eukaryotes in the nucleus. So chromosomes are present in nucleus. So during reproduction, each parent passes one set of chromosome to their offsprings. This means that offsprings receive half of their genes from one parent, that is mother, and half from their father. These genes determine the inherited characters such as eye color, blood group, and even in plants, the flower color, seed color, and many characters. So because of inheritance, offsprings usually resemble their parents, but they are not identical. So it is our common observation, although the babies of human, they are human, but they are not like their parents. They have some variations. So this is the contradiction. In case of inheritance, in case of genetics, we deal with the inheritance that the humans are humans, offspring, they are humans, but they are different from their parents. So these are the slight variations. Both are due to genetic material. So since they receive a unique combination of genes from their parents, so that's why there is variation. So this transfer of genetic information ensures the characteristics that are passed on from one generation to next, maintaining the continuity of life within the species. So the second subtopic is define allele as an alternative form of a gene. We know that in case of organism having two sets of chromosome, that is diploid, there are two uh, chromosomes on each on both the chromosome. A particular segment of gene is there, but the alternate forms of that gene, they are allele to one another. So if the alleles are same, then the individual is homozygous. If the alleles are different, then heterozygous. So this concept will come later on. An allele is an alternative form of gene that is found at the same position, that is locus. So locus is the position of chromosome at a particular level where the gene is present, and for a particular gene, the alleles are there. So each gene may have two or more alleles which control the same characteristics but can produce different versions of it. But in case of diploid organisms, even for a particular gene, if there are more than two alleles, but only two alleles can be present at a particular time, means simultaneously in one individual. For example, a gene for eye color may have one allele for brown eyes and another for blue eyes, if we talk about only eye color. In humans and many other organisms, each individual inherit two alleles from for each gene, one from one parent. This combination of alleles determine how a particular characteristics is expressed. So third subtopic is to understand and use the terms dominance, recessive, phenotype, genotype, homozygous, heterozygous. So these are the basic concepts in classical genetics, and even they are also related with the modern genetics. So what is dominant? A dominant allele is one that shows its effect on the phenotype, that is the physical appearance of character, even it is present if it is present in single copy, that the other allele is of different type, even then that allele can be dominant, can express itself. and it masks the effect of other allele, which is recessive, represented by capital letter most of the time. If we use symbol, if we use letters for a particular genotype or gene. Example, in pea plant, the allele for tallness is most of the time represented by capital T is dominant over the allele for dwarfness or shortness, which is small t. Recessive. A recessive allele only shows effects on the phenotype when two copies mean that individual should have two, both the copies are of same allele. So that is when the individual is homozygous for that allele, it is masked by the dominant allele. It is its feature represented by most of the time small letter, in above case small t. Example, the short or dwarf plant, small t appears only when no dominant capital T is present. Phenotype. Phenotype is the observable characteristics or appearance of an organism resulting from its gene and sometimes even environment. So, example, tall or short plant, brown or blue eyes in individuals or humans or any other animal. So, what is genotype? Genotype is the genetic makeup, genetic base of an organism. The combination of alleles carries for a particular characteristics. For example, capital T, small t are genotypes for the height in pea plant. Homozygous. If an organism is homozygous, when it has two identical alleles for a particular gene. When both the alleles present on homologous chromosome, they are of same type, representing a particular gene, then this organism is homozygous. Example, capital T, homozygous dominant, small t, homozygous recessive. Heterozygous, an organism is heterozygous when it has two different alleles for a particular gene. For example, capital T, small t. The dominant allele will show its effect that the individual will be having phenotype tall. Then the fourth subtopic is use the genetic diagram including Punnett square to predict the results of monohybrid crosses and to calculate the phenotypic ratio, and only required 1:1 ratio and 3:1 ratio, not dihybrid cross, not trihybrid cross. This is only at O level, only one character at a time, which is monohybrid. A monohybrid cross is between two parent organisms which are true breeding for two distinct forms of one trait. Trait is one, but there are two contrasting features, characters. For example, flower cover, color, red or white, and many other. Mendel tracked each trait through two generations. Uh, in case of this terminology, P generation is the parental generation. And most of the time, if we start with pure breeding plants, so this generation is pure breeding. F1 generation is the first generation of offsprings in the breeding experiments, and it is always hybrid. F2 generation is the second generation of offsprings in the breeding experiments, in which there is selfing of F1 into F1. That is, the individuals of F1 generation, which is hybrid, they show selfing between them. There will be a cross between them to get F2 generation. Mendel performed reciprocal crosses, that is, pollen of tall plant to stigma of short plant, and vice versa. If the character is present on autosome, then there will be no different effect. He found that F1 plants resemble only one of the parent, mean dominant one. If we talk about dominant allele, characteristic of other parents reappeared in about 1/4 of F2 plants, but 3/4 of offspring resemble the F1 plants, which was hybrid and showing the dominant character. Mendel saw that these 3:1 ratio uh, possible when F1 hybrid obtained two factors for each trait, one being dominant and other recessive. Factors separated when gametes were formed, a gamete carried one copy of each factor, and random fusion of all possible gametes occurred upon fertilization. Uh, it is notable that Mendel was not knowing the name of gene, so he used the exact word elementon, which is effector. So results of these experiments led Mendel to develop first law of inheritance, which is law of segregation, which states that each organism contains two factors for each factor. Factors segregate in the formation of gametes. Each gamete contains only one factor for each trait. Fertilization gives each new individual to have two factors for that particular trait. So what is Punnett square? Punnett square was developed by much after the Mendel's experiment and is currently being used to explain the studies performed by the Mendel. The concept was introduced by R.C. Punnett in 1905 that was eventually used as a visual representation of Mendelian inheritance mean classical genetics. A Punnett square is simple grid used to predict all possible genetic combinations from a cross between two parents. It shows how alleles from male and female gametes combine to form offspring uh, to produce offsprings having genotype. So this is this is a figure showing this information. For example, P, the parent generation, P, the pure breeding plants. So pure tall versus cross between pure dwarf, pure tall and pure dwarf. So these are pure breeding generations. So what was their genotype? Capital T, small t, small t. Types of gametes, only capital T, and in this case, only small t. So there is a tradition in genetics. So whenever we have a symbol encircled, so it means we talk about and we are considering a particular gamete only at that time. F1 generation monohybrid, capital T, small t. It's a square and having both the alleles. So it is a hybrid and tall. And types of gametes, if we make selfing, so F1 generation, selfing will be able to produce two gametes, capital T, small t, because this individual is having both the alleles. So in case of Punnett square, this is Punnett square. We have the female gametes, capital T, small t, male gametes, capital T, small t. It may be reciprocal one, and F2 generation in case of these squares having genotype of F2. Capital T, tall, capital T, small t, phenotype will be tall, capital T, small t, phenotype will be tall, and small t, the phenotype is dwarf. So Punnett squares are checkerboard is there. So Punnett square is a very easy method to actually give us the visual prediction of a particular cross. So what was the phenotypic ratio in that case? Mean F2 cross, 3 tall, 1 dwarf, 75%, 25%. And genotypic ratio in F2, pure tall, 1 capital T, small capital T, and 2 hybrid, capital T, small t, and pure dwarf only, 1 and small t, small t. In my opinion, when we say law of segregation, this is the clear-cut evidence that there was a segregation of allele at the time of gamete formation, because in F2 there is appearance of a dwarf which is 25% and mean 3:1 ratio. So it is a clear-cut evidence and the result of law of segregation in a cross. Then comes the fifth subtopic rather, explain why observed ratio often differ from expected ratio, especially when there are small numbers of offsprings. Observed ratios often differ from expected ratios, especially when the number of offsprings is small because of chance or random variation in how alleles combining during fertilization. So expected ratios are theoretical, they are not real one. Expected ratio like 3:1 or 1:1, 1:1 when we want to make a test cross between the hybrid individual, with one of the recessive individual, which is pure breeding, to get the information that whether the phenotypically dominant individual is homozygous or heterozygous. If it is heterozygous, then we will be having 1:1 ratio in the offsprings. So it is a test cross. So expected ratios are predicted results based on probability. These they assume that a very large number of offsprings are produced so that all possible combinations of alleles occur in correct proportions. Random fertilization causes variation. Yes. In a real life, fertilization is a random process. Each offspring um, is formed by chance when it means when the male gametes sperm fertilizes with eggs cannot be controlled. So, especially in a small group, the actual combination may not perfectly match the predicted probability. What does it mean? It means that if a sperm is having the all the alleles in that, but by chance if it is damaged or not be able to fertilize, so the result may be different if the number of individuals, number of population is small. So small numbers exaggerate differences. When only a few offsprings are produced, even a small difference in numbers causes a large deviation from the expected ratio. With large numbers, random differences tend to even out and observed ratio come closer to expected values. So it means that in order to make the clear observation, in order to get the exact benefit of the experiment, we should have large number of individuals to eliminate the chances of error or the natural happenings. So example, if monohybrid cross predict 3:1 ratio tall into versus short, so with 1,000 individuals offsprings, we might see about 750 tall and 250 short, close to which is actually 3:1. But with the 20 offsprings, you might get 14 tall and 6 short, so not exactly 3:1, just because of the chance. So in summary, observed ratio often differ from expected ratio because fertilization is a random process, and with small number of offsprings, chances of variation have a greater effect on the results. So then the sixth subtopic state that two identical homozygous individuals that breed together will be pure breeding, the concept of pure breeding in it. Two identical homozygous individuals that breed together will be pure breeding because all their offspring will have the same genotype and show the same phenotype as that of the parents. What is the explanation? A homozygous individual has two identical alleles for a particular gene, for example, capital A, or small a. When two identical homozygous parents are crossed, mean the capital A into capital A, yeah, or small a into small a. All gametes carry carry the same allele, so all offsprings receive identical alleles from both parents, producing offspring that are genetically uniform. They will always breed true. So hence such a cross is called pure breeding cross if the individuals are homozygous. Seventh subtopic is to explain codominance by a reference to inheritance of ABO blood group phenotype A, B, AB, O. So the genes are responsible for that, alleles for that. Capital A, I for immunoglobulin, uh, I capital A mean dominant one, I uh, capital B mean the for antigen for B, and IO, this immunoglobulin is not having any antigen on it. So definition of codominance. Codominance occurs when both alleles of the gene are expressed equally in the phenotype of the heterozygous individuals. Neither allele is dominant or recessive, both show their effect. Mean that in case of hybrid individual, both are equally expressing themselves and phenotype is entirely different. It is a different concept as compared to the incomplete dominance, which is not the part of your syllabus, O level syllabus, in which there is blending. of two extreme. For example, red and white color, uh, the the intermediate will be pink, which is the intermediate between them. But in case of codominance, there is no intermediate. The hybrid is having entirely a new phenotype. ABO blood group system. In humans, blood group is controlled by one gene that has three alleles. I capital A produces antigen A on red blood cells, IB produces antigen B on red blood cells, and IO produces no antigen at all. So, allele relationship, IA and IB are codominant to one another. When both are present, both antigen A and B are produced, and IO is recessive to both IA and IB. So, genotype and phenotype in this case, genotype capital A, IAAA or I not O, the phenotype will be A. Only antigen A is present. If this is homozygous condition, this is heterozygous condition. In this case, IA, IB or IB, IO, the phenotype will be B. Only antigen B is present on the red blood cells. IB, IA, IB, only one possibility, and blood group will be AB. Phenotype will be AB. Both antigen A and B are produced. It means there is the depiction of codominance in that case. IO blood group will be O, no antigen present on red blood cells. So, what is the summary statement? In ABO blood group system, IA, IB are codominant, meaning both are expressed equally in individual with genotype IA, IB, resulting in blood group AB. The IO allele is recessive, producing blood group O only when two IO alleles are present, mean homozygous. Then comes the eighth subunit. Describe the determination of sex in humans, XY and XX and XY chromosomes concept, chromosomes and sex. Humans have 46 chromosomes in each body cells, arranged in 23 pairs. 22 pairs are called autosome because they are not sex chromosomes. One pair is the sex chromosome because it is determining the sex or the gender of the individual, male or femaleness. Sex chromosomes, females are having two X chromosomes, which are XX, and males are having only one X and one Y chromosome. So, what are the gametes? The female are being producing eggs that all carry X chromosomes, and male producing sperm having half of them X chromosome and half of them Y chromosome on that. So, fertilization and sex determination. When fertilization occurs, if an X carries sperm, fertilizes the egg, the zygote will be XY. The resultant offspring will be female. If Y carrying chromosome fertilizes the egg, the zygote will be XY, which is male. Conclusion: the sex of human is determined by which type of sperm fertilizes the egg. The father's sperm therefore determine the sex of the child. Example, summary: X sperms, X and Y. There are two types of sperms. Only one type of egg. Offspring genotype XX, XY. This will be a female, and this one will be male. Then the ninth sub topic: describe a gene mutation as a random change in the base sequence of DNA using the sickle cell anemia as an example. In case of O level, there is only one example, which is sickle cell anemia. The shape of the cell is sickle, and anemia is the disease or disorder. Definition of gene mutation, which is very important. A gene mutation is a random change in the base sequence of DNA. This change can alter the sequence of amino acid in a protein, and which may change its structure and function of protein. How mutation occurs? Mutations can happen spontaneously during DNA replication, or they may be caused by the mutagens such as radiations, X-rays, UV rays, certain chemicals, this will be covered in the next topic. Example, sickle cell anemia. Sickle cell anemia is caused by the gene mutation in the gene for hemoglobin, the protein in red blood cells that carry the oxygen. The normal DNA-based triplet codes for the amino acid glutamic acid, but by a single base substitution, mean the replacement of one nucleotide with the other one, changes its triplet. So triplet mean the three nucleotide in a sequence. So the new code will be valine instead, and the shape of the whole protein molecule consisting 574 amino acid will be changed into a sickle shape instead of normal globular shape. So effect of mutation, the altered hemoglobin called hemoglobin S means sickle shape, makes red blood cell change shape from round to sickle shape when oxygen levels are low. So these sickle cells can block blood vessels, causing pain in tissues. They also break easily, leading to anemia, shortage of red blood cells, and the carrying oxygen carrying capacity will be affected very much in quantity. Summary of it, a gene mutation is a random change in the base sequence of DNA. In sickle cell anemia, this mutation alters the structure of hemoglobin, causing red blood cells to become sickle shaped, leading to health problems. So the 10th subtopic, describe a chromosome mutation as a change in chromosome number or structure using Down syndrome as an example, which is having 46 chromosome instead of 47 chromosome instead of 46. So there is only one Down syndrome in our O-level syllabus. Definition of chromosome mutation. A chromosome mutation is a change in the number or structure of chromosome in a cell. This type of mutation affects whole chromosome rather than individual genes. Type of chromosome mutation. Chromosome mutation may involve change in number, having extra or missing chromosome, or change in structure. Parts of chromosome may be lost, duplicated, rearranged, or what we say, the position change. So example of the given example is Down syndrome. Down syndrome is caused by change in chromosome number. So normally humans have 46 chromosome, 20 into 23 pairs, but individual with Down syndrome having 47 chromosome. This is due to an extra copy of chromosome number 21, a condition called trisomy at 26 chromosome number level. How it is occurs? During the formation of gametes in meiosis, when there is the synapses or combination of homologous chromosome, sometimes the chromosome fail to separate properly. This is called non-disjunction. So the result will be one gamete may receive an extra chromosome of 21. When it combines with the normal gamete at fertilization, the zygote ends up with 3 copies of the chromosome number 21, and total number of chromosome in that individual may become 47 instead of 46. So the outcome and effect of Down syndrome, people with Down syndrome may show characteristic facial features. Their face is quite obvious, and anybody can guess that this person is having Down syndrome disorder. Learning difficulties, heart and other health problems. So chromosome mutation is a change in number or structure of chromosome. In Down syndrome, this occurs due to extra chromosome extra chromosome in uh, chromosome number 21, giving 47 chromosome instead of 47 instead of 46. Then the second last subtopic, state the mutation, meiosis, random mating, and random fertilization are sources of genetic variation in population. So if we talk about why the population mean the presence of individuals of the same species, they are different from one another. The causes are mutation, produce new alleles by changing the DNA base sequence, creating new genetic information. So we always say that mutation is the raw material of genetic variability or evolution even. Meiosis introduces variations through independent assortment of chromosome and crossing over between homologous chromosome. So meiosis is only giving the reshuffling of genes, reshuffling of alleles, and the crossing between them, but the actual raw material is mutation. Random mating ensure individual by chance allowing different combination of allele to occur. There is no discrimination. So random fertilization, any sperm can fertilize any egg producing unique combination of gene in offspring. So these are the reasons for the change in population. So these processes ensure each individual in a population is genetically different, providing the genetic variation on which natural selection can occur and can act even. The last subtopic of this subunit inheritance, understand that ionizing radiation and some chemicals increase the rate of mutation. Rate of mutation. Mutation is there, but rate of mutation may become increased. Ionizing radiation and certain chemicals can increase the rate of mutation. Explanation: normal mutation rate happens, the mutation may happen to occur spontaneously, mean by nature, by themselves, and rarely during DNA replication. Mutagens, certain environmental factors can increase how often mutation occur. These are called mutagens, and example of mutagens are given. Ionizing radiation such as X-rays, gamma rays, and ultraviolet rays can damage DNA and can cause the change in base sequences, so mutation. Chemical mutagens such as tobacco, smoke, mustard gas, and some industrial chemicals can alter the DNA or interfere with its replication. So possible effect, some mutations are harmless or silent because if there is no change, although the base change in base, but the code for a particular amino acid will remain same, then there will be no effect. Some may cause genetic disorder or cancer if there is change in genetic code. Occasionally, mutation may be beneficial, very rare, contributing towards evolution. What does it mean? That if by chance, if there is a change in base sequence and the functioning of the resultant protein become more efficient, then this change will be, this mutation will be beneficial. So ionizing radiation and certain chemicals act as mutagen, increasing the rate at which mutation occur by damaging or altering the DNA in cells. Ionizing radiation chemical increase mutation rate. Mutation is random change in base sequence of DNA, while mutation can happen naturally. Some factors increase their rate, and these factors are mutagens. Ionizing radiations, X-rays, gamma. This is repetition of our previous discussion, but with slight change. So these ionizing radiation can break DNA strand and alter bases in the DNA molecule. When DNA is damaged and not properly repaired, mutation may occur in a gene or chromosome. High exposure to ionizing radiation can therefore increase mutation rate and sometimes lead to cancer or genetic defects. So example, X-rays used in medical imaging, large or repeated doses, although the patient and the individual medical paramedical staff or medical staff, they are being exposed to X-rays calculatedly, and there is a limit to get exposure to that X-rays during their working. Cosmic rays, we don't have control over them, and they are coming from outer space. Nuclear radiation, yes, we have control, and this is new phenomenon in our observation. So they are from radioactive materials. Chemical mutagens, some chemicals can also cause mutation by changing the structure of DNA or interfering DNA replication. These are often found in industrial substances, cigarette smoke, or polluted air. Example, tar and nicotine in the smoke. Mustard gas and other industrial chemicals, pesticide and food preservatives in high doses. So there is a summary mutagen type, its example and effect on DNA. Ionizing radiation, X-ray, gamma rays, UV rays, breaks or alters DNA strand. This will be the effect on DNA. Chemicals, tar, nicotine, pesticide, alter base sequence or replication process. So this is all about the topic sub unit inheritance, in which we actually concerned with the genes and outcomes of the individuals. So this topic is quite lengthy. So thank you for bearing me up. Allah Hafiz.